As I say, good morning, everybody. My name is Trevor Dummer. I'm the National Scientific Co-Director of CanPath and also the Co-Scientific Director of the BC Generations Project. Thanks for joining us here today for our webinar. Very excited to bring together 3 research trainees who've been using CanPath data and CanPath samples in their doctoral research. And one of the objectives of CanPath has always been, right from the outset, to create a platform to support research for the next and training of the next generation of scientists. So it's really exciting to bring together 3 doctoral candidates who are utilizing the various regional cohorts, parts of CanPath, for their doctoral studies. So before I introduce the speakers and we could get on with the actual presentations, just a couple of items of housekeeping. So we've got 3 presentations, so we're going to have to stick very closely to time, 10-minute presentation for each. We do have some time for questions, so please use the Q&A button to type your questions in. We'll try and get to all of them, but each speaker only has around about 5 minutes for questions. But if you type your questions in, and if we don't get to them, we'll certainly be able to try and answer them over email. And you can also keep in contact with CanPath through the info@canpath.ca email address. Those of you that are on Twitter can follow us. Twitter handle is WeAreCanPath. People on LinkedIn can look at CanPath as well. So before we move on to the presentations, I would just like to acknowledge that today I'm located on the traditional unceded territory of the Musqueam, Squamish and Tsleil-Waututh peoples, and we thank them for allowing us to reside as settlers on their traditional territories. Also, we're meeting together today on a virtual platform, so we'd like to also acknowledge that, wherever we are, we're on the ancestral and unceded territories of the Inuit, the Métis and First Nations people that call this home. Okay, so before we move on to the 3 specific speakers, I just want to provide a brief bit of background to CanPath, just so that you know exactly what data we have available and what resources are available. Great. So as you can see from this diagram, CanPath, it's a project that's been about 10 years in the making now. It's Canada's largest national longitudinal population health cohort. We've recruited over 300,000 individuals, and the aim is to follow these individuals over the next 50 years. So it's a truly longitudinal cohort. It's founding cohort members from East Coast to West Coast of the Atlantic Path, covering the Atlantic provinces, Cartagena, which covers Quebec, then the Ontario Health Study, and then Alberta's Tomorrow Project, and finishing up here on the West Coast with the BC Generations Project. CanPath is now truly covering all the provinces of Canada. Manitoba Tomorrow Project has recently started recruiting, and I'm pleased to say that the Saskatchewan Partnership for Tomorrow's Health has a pilot approved and will be moving hopefully to full recruitment soon. So 300,000 adults being followed over the next 50 years. And we've captured a whole broad range of data related to obviously health and lifestyle and risk factors for disease. And the idea was to capture broadly data that can help us understand the causes of initially cancer, but also really it's about other chronic diseases as well, because obviously the causes of cancer, and we were originally have been funded by the Canadian Partnership Against Cancer. So it was built as a cancer cohort project, but it's very much a chronic disease cohort project. At baseline, we've captured a wealth of health and lifestyle data through a core baseline questionnaire, and that was administered to all 300,000 individuals. We've also got the opportunities to link to administrative health data. All participants have consented to record linkage. We've captured biological samples. I'll talk about those shortly. We've weighed and measured people where we could, so we've captured physiological data. We've been able to link to environmental data through the National Canoe Project as well. So very much the idea is about linking individual behaviors, environmental data, health systems data, health-seeking data, biological information, linking that all together to try to really understand some of the causes of infection, causes of disease. I mentioned biological samples. So the objective of CanPath was always to capture a venous blood sample from as many of the participants as possible, and we've managed to put a needle in an arm of nearly half of the cohort population. So we have a venous blood sample from nearly 150,000 individuals, urine from almost a third of the cohort as well. Saliva, one of the speakers will be talking about using some saliva samples later on a smaller cohort, but we have a smaller number of the cohort, but we have some saliva samples. Blood spots were captured from individuals living in remote and rural locations who were unable to come to us, or we were unable to go to them to capture a venous blood sample, but the blood spot can be returned in the mail. So we were able to capture and include biological samples from people from quite remote locations. Atlantic Path was the only regional cohort to capture toenail clippings, and one of the speakers will be talking about the use of the toenail clippings later. And anyone that's ever heard me talk at these sorts of things always reminds people that Atlantic Path, and that's where I actually started my work with CanPath. Atlantic Path holds the Guinness World Record for the largest toenail collection. Okay, so it's a longitudinal cohort. We've been engaged in very active follow-up as well as passive follow-up. So passive follow-up is through linkage to administrative health data, cancer registries, and other electronic means of data capture related to health. But active follow-up is through recontact. We completed the first wave of follow-up questionnaires a couple of years ago now, and 120,000 participants completed a very detailed health and lifestyle questionnaire, which included information that we asked at baseline is within the core questionnaire, so it was repeat measures on that, plus some additional information related to behaviors such as marijuana and e-cigarette use. The whole range of fairly standard health and lifestyle information, which provides us with very valuable information over the life course, really, because this is longitudinal data now. Now, CanPath was originally established as a chronic disease cohort and a cancer cohort, and I don't think many of us really envisage there will be much use or a great deal of use of the cohort for infectious diseases. But COVID came along, as we all know, and earlier last year, and we quickly pivoted to see whether the resources, the CanPath resources, could actually be put to valuable scientific use to help combat this virus. So we launched the CanPath COVID-19 questionnaire. We've managed to seek, be grateful for support from CIHR and from the Canadian Immunity Task Force to actually do a questionnaire and capture some more biological samples and actively research some of the risk factors associated with severity, for example, of COVID-19. We've captured information on mental health, job stress, social, financial well-being, all of those sorts of things. Sources of exposure, and trying to really, hopefully some of the information we have will get a sense of why some people do actually have a much harder time with the virus than others, and why some people are more at risk than others. So the COVID questionnaire was completed by just over 100,000 individuals, and I'm happy to say that that questionnaire was certainly the fastest questionnaire development and implementation I've ever been involved in a cohort study. I'm happy to say that questionnaire information is now actually available through an expedited access process, and I'll talk about access in a moment. So a longitudinal cohort really is all about building up and linking information in real time, so you get a history of exposures and a history of behaviors, and then you get health outcomes at certain points in time. And within observational epidemiology, we all know that the longitudinal cohort is one of the best means to actually try to understand disease causation. So we have the potential for a lot of record linkage. We've already linked to the built environment for environmental data. Many of the regional cohorts are actively involved in linkage to cancer registries, for example, and one of our speakers will talk about the research that's been supported by linkage to cancer registries. So we have the means to link the baseline and follow-up information from a whole raft of administrative and other data to really, truly, as I say, build up a picture of health risk over the life course. Very briefly, before I introduce our first speaker, just to mention that there is a data access process. This is a platform for you as investigators to utilize, so I would encourage everyone to look at this, go and visit the data platform, get a sense of what data is available and what the access process is. And these slides will all be available earlier, so you don't have to frantically write down these websites, or indeed if you just put into your favorite search engine CanPath, Canadian Partnership for Tomorrow's Health, you will find these resources pretty quickly. Okay, so all I really need to do now is just thank all of the participants in CAMPATH. They've really made this cohort the success that it is, and the success that it will be as we move over time. I'd also like to acknowledge the sponsors and funders of the project. Okay, so that's all I wanted to say. I'm now going to hand over to Molly. I think I need to stop sharing my screen, but I can't find the button. So there it is. Right. So while Molly's getting herself set up, I'll be able to just briefly introduce her. So our first speaker is Molly Sweeney McGee. Molly is a PhD candidate at the School of Population and Public Health here at UBC. So Molly's also a public scholar. So part of her work really is aiming to try to improve how you apply academic research into the real world to solve public health issues. Molly's work has used the BC Generations cohort data, longitudinal follow-up questionnaire, and linked cancer data to look at the health behaviours in colorectal cancer prevention and survivorship. Now, prior to coming to Canada to pursue her PhD, Molly worked as a research associate in health behaviour change at the University College in London. And her primary interests are in cancer prevention, healthy aging, behavioural interventions, and implementation fidelity. Okay, so over to you, Molly.
Thanks, Trevor. So as Trevor mentioned, I'm just going to talk today briefly about one part of my PhD research, which was an examination of the impact of a cancer diagnosis on certain health behaviours. So to conduct this study, I was able to take advantage, as Trevor mentioned, of the prospective nature of the BC Generations project and look at change in these health behaviours before and after a cancer diagnosis. So I'm just going to run through the reasons I decided to focus on this topic, the specific aim of this research, and I'll briefly describe how the BC Generations data were used, the results we found, and the implications of our findings in terms of future research. So thankfully, improvements in early detection and treatment have led to an increasing number of people surviving cancer. However, this expanding group of cancer survivors tend to be in poorer health than the general population, and they have increased risks of future additional primary cancers, as well as comorbidities like cardiovascular disease and diabetes. And the evidence shows us that poor health behaviors like being overweight, smoking, and being inactive are really important determinants of health outcomes in cancer survivors. So for example, they've been shown to be associated with cancer recurrence, cancer-related mortality, and total mortality in cancer survivors. I also wanted to focus on behavior change after a cancer diagnosis, because it's been suggested by a number of researchers that the gravity of a cancer diagnosis could work to motivate lifestyle change and also be a teachable moment for improving health behaviors through behavior change interventions, because people might be most open to changing their behaviors at the time of a cancer diagnosis or perhaps shortly afterwards. However, I found in the literature that the evidence on the actual impact of a cancer diagnosis on health behaviors is quite sparse. In the research that does exist, there tend to be some methodological limitations. So most studies are cross-sectional. They rely on retrospective reporting of behavior by cancer survivors prior to diagnosis, and even those that do have longitudinal data, the pre-diagnosis health behavior data tend to be gathered retrospectively after diagnosis. So with these sort of designs, obviously we're worried about recall and reporting biases. So to try and address this paucity of research and the methodological limitations in the literature, we decided to conduct a prospective analysis of health behavior change in survivors of one cancer, in particular colorectal cancer, by using longitudinal data from the BC Generations project. So given that the demographic characteristics, the treatment side effects are quite unique to individual cancers, we decided to focus on assessing behavior change for one common cancer, colorectal cancer, although we did include of cancer survivors of other cancers as a separate group in our analyses. So our aim was to describe and model health behavior change between or to describe, to describe and model health behavior change between baseline and follow-up for participants diagnosed with colorectal cancer and other cancers relative to those who remained cancer-free between baseline and follow-up questionnaire time points in the BC Generations project. So for the analysis, I included any participant with data available at both baseline and follow-up. I then excluded participants with prevalent cancer at baseline, again, using the linkage with the BC Cancer Registry, which left me with 19,764 participants remaining for analysis. And the mean time between baseline and follow-up questionnaire completion was 5.3 years. So we were able to gather sociodemographic data from both baseline and follow-up, and cancer diagnoses were established through the linkage that Trevor mentioned between the BC Generations project and the BC Cancer Registry. The health behaviours that were of interest and available in the data were average daily alcohol consumption, smoking status, and BMI. And of course, these were gathered at both baseline and follow-up questionnaires, so we could look at change over time. So in terms of the analysis, we found firstly that there were very few smokers, so we didn't include this group in our models in the end, and I'm not going to discuss it further today. But in terms of grouping, as I mentioned, participants were divided into 3 groups: those diagnosed with colorectal cancer in the interim between baseline and follow-up, those diagnosed with any other cancer in that same time period, and then those who remained cancer-free between the 2 time points. In addition to producing some descriptive statistics for each of these groups, I modeled both health behaviors, BMI and average daily alcohol consumption at follow-up using multiple linear and logistic regression to look at differences between baseline and follow-up for participants diagnosed with colorectal cancer or any other cancer relative to those who remained cancer-free in the interim. And so these models adjusted for baseline values for the health behaviors in question, as well as confounders in a stepwise manner. So in terms of results, we found that 742 participants received a cancer diagnosis, excluding non-melanoma skin cancer, between baseline and follow-up in our sample. Of these, 63 were colorectal cancer cases, which was 8.5% of the sample of participants with cancer. There were small changes across groups in BMI and alcohol consumption over time. So for example, here you can see with BMI that there was a small reduction in average BMI between the 2 time points, and this change was similar for all 3 groups. In the adjusted analyses, the only significant change was for BMI in participants with cancer other than colorectal cancer relative to the cancer-free group. Cancer status was not associated with change in alcohol consumption between baseline and follow-up for the analyses, comparing colorectal cancer survivors and other cancer survivor groups to the cancer-free group. So just briefly, I think the research identified a trend for decreased BMI between baseline and follow-up across all participants in our sample, but there was no significant change between baseline and follow-up for BMI and alcohol consumption for the colorectal cancer group relative to the cancer-free group. In contrast, participants with cancer other than colorectal cancer had small but significant reductions in BMI from pre to post-diagnosis relative to the cancer-free group. So this finding of lack of change in colorectal cancer survivors specifically is supported or supports the only other prospective analysis we could find in the literature, which was one conducted in 2009 by Ski and colleagues. They also found no change in BMI and alcohol consumption for colorectal cancer survivors before and after diagnosis. So I think our research is suggesting that colorectal cancer survivors are not changing their health behaviors after being diagnosed. Similarly, while other other cancer survivors did reduce their BMI, the changes are very small and perhaps of limited clinical significance. So in terms of future research, it would be good to repeat the analyses that we've conducted here in larger samples, perhaps in other CANPATH cohorts, and also at future BC Generations project follow-ups, as ongoing health behaviors after diagnosis would be a much stronger predictor of health outcomes than just those behaviors at one time point, as we were able to measure here. I think it would also be useful to examine some other health behaviors that have been shown to be important in determining colorectal cancer survivor outcomes. so especially physical activity, which has actually been most strongly linked to colorectal cancer survivor health outcomes. Strengths of our study include its prospective design, allowed for by the nature of the BC Generations project, and the objective assessment of cancer diagnoses, again allowed through the linkage between the BC Generations data and the cancer registry. However, we had some limitations to the number of participants. Obviously, in the colorectal cancer group was low, meaning our results have to be interpreted with caution. There were also some missing data for the alcohol variable at baseline, although that was mostly due to differing questionnaire versions. So it's likely that the data were missing completely at random and thus unlikely to have introduced bias to our analyses. So in conclusion, I think our results tentatively suggest that both colorectal cancer survivors and all cancer survivors are making limited changes to their health behaviors after diagnosis, despite suggestions that such a diagnosis could prompt these changes. So as such, I think that these groups could really benefit from targeted health behavior change interventions to improve these really important lifestyle factors. But some additional work is needed to clarify the reasons for this lack of change identified here, and to really better inform the design of future health behavior change interventions that could help these groups. And that's everything. I'd like to acknowledge my supervisor, my supervisory committee, the salary and funding support I've received, and also the BC Generations Project staff, and of course all the participants. So yeah, thank you.
That's great. Thank you, Molly. So I don't see any questions in the Q&A at the moment. So as I mentioned, you should be able to see the Q&A box, I believe, at the bottom of your screen, or maybe it's on the side for me. So please do, if you have any questions for Molly, type them into there. I guess seeing none, I've got a very quick question actually, Molly. So in terms of the change in BMI, how much do you think that's to do with potentially a change in behavior and a positive change in BMI for those reasons, or to do with treatment?
Yeah, that's a good point. I think if I was doing it again, or if we had more cancer survivors to look at, it would have been interesting to maybe limit our analysis to look at people who were 6 months or more, maybe on from finishing treatment or on from diagnosis. So I think it would be clearer that what we'd be seeing was due to actual lifestyle change rather than side effects of the treatment. But yeah, again, I think we just didn't have the numbers to do that, especially with the colorectal cancer survivor group. But yeah, that definitely could play a part. But yeah, I think it would be interesting to look at in the future, hopefully.
Yeah, and actually, the final sort of quick question from me as well, I guess. So, and if the evidence from this, obviously, and this was a smaller study, but some evidence from elsewhere shows that there's not a huge amount of behavior change. Why do you think that is? I mean, if do you have any, because you've been very interested in why people change behaviors. People obviously, it's a cancer diagnosis and cancer treatment, they're coming into contact with health professionals. Why what's the barrier, do you think?
Well, I think they're not getting the sort of intensive support that is probably needed, and they've obviously survivors have probably got a lot of health concerns and concerns in their lives in general, and I think it's a time where they need a lot of support. And I know from looking at the literature that sort of healthcare professionals maybe find it difficult to make those to be that sort of support to them at that time point, because there are mixed messages that they get to in terms of maybe they want to encourage cancer survivors to sort of build themselves up and maybe put on weight to sort of help them recover from treatment. So I think the messages are quite difficult for people who maybe haven't received training in health behavior change interventions. They're quite difficult for them to implement. I think that's why it's important that there be targeted programs led by people who have the training in these sort of skills to really be effective.
And I see a couple of questions popped up, and I realize we should be moving on to Jacob a couple of minutes ago. We did buffer in a couple of minutes, so I'll maybe ask the first question, then we'll come back and hopefully we'll be able to get to the second question as well. So is it possible BMI wasn't a good indicator and that another anthropometric measure may be better, such as waist or hip? So did you try that?
Yeah, I think we focused on BMI just because it sort of is the classic measure in the literature. It tends to be what people focus on. I believe I don't think we had another measure that I could have used. I could definitely look again at our data, but I'm pretty sure BMI was the main one that we had, and especially the one that we had from the most gravest amount of people, because it was a self-report measure as well as nurse measured in some cases. Whereas I think with the other measures they were just nurse measured, so we wouldn't have them for as many people.
Okay, great. Thank you very much. So, Jacob, a couple of minutes behind schedule, but you're up next. So, if Molly will stop sharing her screen and you can switch your camera on and start sharing your screen while you're getting ready, I'll just do a very brief intro to Jacob. So, Jacob Nearing is a PhD candidate at Dalhousie in the Department of Microbiology and Immunology. Since joining graduate school, Jacob's been pretty prolific. He's published 5 papers, including 3 first author papers, and presented at a number of national and international meetings, and won a poster award at the International Human Microbiome Consortium in 2019. So Jacob's research interests include the interactions between cancer and the human microbiome, bioinformatics, virology, and data science. And he's currently funded through various scholarships, including Nova Scotia Graduate Studentship Scholarship and the Research Nova Scotia Scholars Award. So over to you, Jacob.
Awesome. So today I'm here to talk about some of the work I've been doing for my PhD, looking at how the oral microbiome is associated with cancer using data from the Canadian Partnership for Tomorrow's Health. So many of you may have heard about the microbiome in the news. It's recently become a very hot topic, being connected to a number of different health issues. And to give a formal definition to the microbiome, it's actually the microbes and their actions within a certain environment. And in the human body, there are a number of different areas you can study. And some of the key areas we've looked at so far are the oral microbiome, the lung microbiome, the skin microbiome, the gut microbiome, and the vaginal microbiome. And all of these different areas have been connected to various different diseases and health outcomes. For instance, the gut microbiome has been strongly connected to the development of inflammatory bowel disease, whereas the vaginal microbiome has been connected to things such as preterm birth. However, today what I'm most interested in talking about is the oral microbiome. And the oral microbiome has been strongly connected to a number of diseases, including the development of gingivitis, as well as various oral cancers, where they've shown that the bacterial community in the oral cavity is significantly different in people with oral cancers versus healthy controls at diagnosis, and has even been strongly connected to heart diseases such as atherosclerosis, where the production of nitric oxide by microbes within the oral cavity actually helps change blood pressure. And while the microbiome has been connected to a number of these diseases strongly, it has also been associated with a number of other diseases. So recent research has shown that the oral microbiome is significantly different in individuals with pancreatic cancers compared to healthy controls at diagnosis. And a number of research studies have shown that the oral microbiome is significantly different in individuals with inflammatory bowel disease. Another cancer that has been linked to the oral microbiome is colon cancer, and it's actually been through a number of different studies, with one study showing that the oral microbiome is significantly different at diagnosis in people with colon cancer when compared to people with adenomas or healthy individuals. And multiple other studies showing that the gut microbiome is actually enriched with microbes normally found in the oral cavity in individuals with colon cancer, suggesting that perhaps the oral microbiome may play some sort of role in colon cancer. And while the oral microbiome has been connected to a number of diseases, it's also impacted by a number of daily life factors. This includes things such as body composition, the types of diet people eat, the lifestyles they do, such as how much exercise they get on regular, the type of infections an individual has, as well as their infection history, the status of the individual's immune system, and various other demographics such as age and sex. And while there have been a number of studies that have looked at all of these things individually, there has a current lack of a study that tries to connect all of these different variables together. And so this kind of led me to the first objective of my project before I even started looking at cancer, which was to determine what anthropometric, dietary, and lifestyle factors are associated with changes in the oral microbiome. And so we answered this question using data from the Atlantic Partnership for Tomorrow's Health project. At the time, they had over 35,000 individuals enrolled. And from those 35,000 individuals, they had roughly 9,000 saliva samples. From these 9,000 saliva samples, we selected 1,200 from healthy individuals that indicated no presence of disease. And we sequenced the DNA within the saliva samples to get an idea of the taxonomic or types of bacteria found in the oral cavity. After DNA sequencing and quality control, we were left with about 1,000 samples. And with these 1,000 samples, we divided them into an initial analysis or exploratory data set and a validation analysis. And in this analysis, we looked at over 41 different variables that might impact oral microbiome composition, including things such as the geography of the person that gave the saliva sample, various demographics, a number of body composition measurements, and dietary patterns. So I don't have time to go through all of the results, but the main results we found were that there are a number of different factors that are related to oral microbiome composition. This includes things such as age, the individual's fat-free mass, as well as the number of refined grains an individual eats. And while all of these variables were significantly related to oral microbiome composition, we surprisingly found that none of them explained more than 1% of the total variation found in the oral microbiome composition, suggesting that they only affected a small number of types of bacteria in the community and didn't cause overall large shifts. And so to look at this a bit further, we looked at which microbes might be enriched for these various factors, and we found a number of associations as shown here. And some interesting ones I'd like to point out are that we found an enrichment of a relatively uncharacterized bacteria in the Diffluviotelliaceae family that was found to increase in abundance as the amount of time passed since an individual's last dental visit. And this was interesting because this bacterial group has recently been connected to the development of chronic gingivitis. We were also able to recapitulate previous results that showed that bacteria in the family of Villanellulaceae are significantly enriched in male individuals. And we even found results that we didn't think would occur. And we found that the abundance of mycoplasma bacteria actually decreased with age, which was opposite to what we would have thought, as mycoplasma infections generally increase as an individual's age increases. And so with that data, we are also interested in looking at potential metabolic changes in the oral microbiome. And we found a number of different metabolic pathways that were related to various daily factors. And so, however, the one that we found to be most related to changes in metabolic potential was refined grain servings. And interestingly, refined grain servings was mostly connected with differences in various carbon pathways, as highlighted here in red. And this is interesting because the oral microbiome is very dependent on the types of carbons individual eats. And so as the different amounts of refined grain servings an individual eats, that will bring different types of carbons into the oral microbiome, which might cause shifts in the oral microbiome to be able to use those types of carbons. And so with that, in summary, we're able to show that a number of anthropometric dietary factors and some demographics were associated with the oral microbiome. However, each of these factors explained relatively low amounts of total variation. Finally, we were able to also see that refined grain usage was linked to potential metabolic changes in the oral microbiome. And so with objective 1 looked at, we are now interested in looking at determining the presence of oral microbiome biomarkers in breast, prostate, and colon cancer. And this is the objective I'm currently working on. To answer this objective, we collected samples from both the Atlantic Partnership for Tomorrow's Health project, as well as the Alberta's Tomorrow project, both cohorts in CanPath. These cohorts provided saliva samples from individuals with breast, colon, and prostate cancer. And all of these samples were actually collected when the individual was cancer-free. So at collection, the individual was cancer-free, and then these samples were then diagnosed through either follow-up data or cancer registry data as having developed cancer after sample collection. So this will give us an idea of possible oral microbiome risk factors for the future development of cancer. And each of these samples will then further matched with a case control style based on sex, age, and smoking. And unfortunately, I've just received this data very recently, so I didn't have time to generate any preliminary results. However, expected or future directions for the project are: 1, to determine potential risk factors for breast, prostate, and colon cancer found in the oral microbiome. 2, to classify individuals that go on to develop cancer based on oral microbiome signatures, so whether or not the oral microbiome is useful as a classification tool. And finally, to connect the oral microbiome with other CANPATH data, some of which we will hear about today. For instance, we're interested in looking at how the oral microbiome may be related to heavy metal exposures from toenail clippings, which you'll hear about in the next presentation, as well as various blood metabolites and genetic signatures from DNA material that's been collected from these participants. And with that, I would like to thank all of my lab members, my committee members, my research funding, as well as CANPATH and the 2 cohorts I've been working with very closely, as they've been very useful for answering all of my questions and providing all the samples. And with that, I'm happy to take any questions.
Thanks so much, Jacob, for excellent, really interesting presentation and keeping perfectly to time. So just while we're waiting for people, if they want, as before, type any questions you have into the Q&A box and we'll hopefully get to them now. If not, we can follow up with them later. I have just a question to start you off. And I guess this is, I mean, I know you're a microbiologist and immunologist. My interest is in cancer prevention. So, and this fascinating findings, how do you see these sorts of findings moving their way through to, I guess, people like me or people like Molly who are interested in behavior changes and preventing disease? So what are the implications for that world?
Yeah, so that's a great question. So like the first part of our study was very interested in looking at basically how behaviors might affect oral microbiome composition to begin with. And so we're hoping to look at whether or not those changes are related to specific types of bacteria that then might be related to an increased risk factor for the disease. And whether or not the microbe is actually playing a direct role in the development of the disease is unsure, but it may be act as a signature to suggest that certain environmental exposures that individual might have might cause the enrichment of that bacteria, which suggests that they might be at more risk for the disease in the future.
Great. Okay, so I'm not going to give you a chance to have a drink of water because we've got 3 questions now, so we'll see if we can get through all of these. So first question from Ethan: can DNA sequencing truly identify every member in the oral microbiome accurately?
Yeah, so that's a good question. And so the type of sequencing we did is based on a small conserved gene found in all bacteria, the 16S ribosomal RNA gene. And so in many cases, we're unable to identify the specific species or strains of bacteria found in the oral cavity, but can generally figure out the genus level of bacteria.
Oh.
So the general idea of what types of bacteria are there.
Right. So Jake, how does mouth washing or toothbrushing affect the microbiome and the saliva samples that you've been looking at?
Yeah, so that's a great question. Unfortunately, we don't have a lot of information on the individual's oral health habits, so we didn't have any information on how often they brush their teeth and things. But there has been a lot of research to show that that after an individual brushes their teeth, the oral microbiome, there are significant changes in the composition of the oral microbiome. However, that being said, there's also been a lot of work to show that the oral microbiome's signature is relatively stable over time. So like the brushing your teeth will cause some bacteria to change in abundance, but it won't change your overall profile that significantly.
Okay. And I guess similarly, if someone's just recently eaten a meal, it might impact.
Yeah, exactly.
Okay, thank you. And the final question before we move on to our last speaker then. So how did you classify dietary data? Did you have a number of dietary items and define some of them, and you define some of them? So how did you classify the dietary data?
Yeah, so that's a good question. So from the data that was collected, there was a number of different variables, and we just used them each individually. So there were categories such as the number of milk servings an individual has, the number of fine grain servings, as I said earlier, as well as vegetable servings, juice servings, etc. There was like at least 20 variables collecting various different things, including things such as like the number of how much spice an individual uses generally on their food, or how much soda pop they drink during a day.
Great. Okay, so that was fantastic. Thank you very much, Jacob. So we'll move on to our final speaker now. So if you could unshare your screen, Callie, if you could start sharing your screen, and while you're getting prepared, I'll just very briefly introduce you. So our third and final speaker is Callie Hood, who is a PhD student at the Centre for Water Resources Studies in Civil and Resource Engineering at Dalhousie University in Halifax, Nova Scotia. So Callie's research is focused on drinking water quality, with a specific focus on lead and nanoparticles in the water distribution system. Callie previously worked at the Public Health Agency of Canada for the Centre for Chronic Disease Prevention and Health Promotion. She received her MSc in Community Health NEPI from Dalhousie in 2020 and a BSc in Physics and Mathematics from Mount Allison in 2017. So over to you, Callie. Thank you.
Hi. Yeah, so thank you for introducing me, Trevor. As Trevor mentioned, I'm currently a PhD student in my first year at the Center for Water Resource Studies at Dalhousie University. And today I'm talking to you about my master's research that was done in partnership with the Health and Environments Research Center Lab and the Atlantic Path cohort here. And the title of my research is Using Toenails as Biomarkers to Compare Arsenic Speciation and Metabolomic Profiles in Breast, Cervical, Prostate, and Skin Cancers. And so like the other presenters today, and probably like many of the people on the call today, the goal of my research is to improve the health of Canadians. And when I think about this question on how we can do this, I think that we can boil it down in a couple of different ways. Like, what makes Canadians sick, and what are the determinants of health? And the Public Health Agency of Canada has outlined a number of different health determinants, and they've been categorized into different levels like your lifestyle factors, healthcare, your individual biology, and your environment. And so your previous speakers have touched on some of these early items, but my focus is really on the final one, the environment, with a particular emphasis on exposure to toxins or heavy metals. And so the main focus of the work I'll be presenting today is on exposure to arsenic, but I did also look at other metals as well. The Agency for Toxic Substances and Disease Registry has classified arsenic as the #1 global health hazard, and other metals like lead, mercury, and cadmium rounding out the top 10. And so when we're thinking about arsenic, we know that it's found naturally around the world in the soil. And from there, it can leach into and contaminate the water, where it's commonly found in its inorganic form. And inorganic arsenic has been associated with several cancers, and the International Agency for Research on Cancer has said that it may cause some others. And drinking water is really considered the main exposure pathway, although there are others, including inhalation and skin contact. But due to the importance of drinking water, the World Health Organization and Health Canada have both recommended that the concentration of arsenic in water not exceed 10 micrograms per liter, So while regions around the globe have varying levels of arsenic in their soil, here too in Atlantic Canada, we have high levels of geological arsenic. In addition to this, Atlantic Canadians rely heavily on private wells for domestic water supply. And when you consider these two factors together, Atlantic Canadians are of higher risk of arsenic exposure. For example, a study in Nova Scotia found that Atlantic Canadians have a very high risk of arsenic exposure in their well water, as depicted here in the dark purple. A large proportion of Nova Scotians are at risk. In addition to this, the Atlantic region has the highest age standardized incidence rates of cancer in the entire country. And so when you consider all these factors together, this region is really ideally suited for investigating any potential associations between arsenic exposure and the development of arsenic-related cancers. And so many of you are probably not familiar, but I'm going to explain briefly the thought behind the link between arsenic exposure and cancer. And so once ingested, arsenic undergoes, like everything else, a metabolism process inside the body. And this process is called arsenic methylation. The research has shown that arsenic methylation produces two different metabolites, mono and dimethyl arsenic species, and the ability for an individual to produce these different species is considered their arsenic methylation capacity. Different species are thought to have different toxicities, with the MMA species being considered the most toxic. However, because different methylation capacities have been observed across populations and between individuals, recent focus of interest in research has been to discover what is influencing that. And so just like we know that there are biological and lifestyle factors that influence one's cancer risk, we think that these factors may also influence one's ability to methylate arsenic. But how do we quantify these associations? And that's really where my research comes in. So thankfully, the Atlantic Path cohort study that Jacob previously talked about, and as well as Trevor, collected a bunch of toenail samples from their participants, in addition to the questionnaire data. And we were able to using a novel approach, the toenails to quantify both the total concentrations of metals in the toenails, but also the different species of arsenic. And this is really considered a novel approach because previous research has used urine, which is considered the gold standard of heavy metals biomonitoring, but it's limited by several factors, including that it's not reflective of a long exposure period. Toenails might be a more relevant biomarker because of their growth, prolonged growth period. They are reflective of up to a year's worth of exposure and may be more relevant for evaluating chronic disease. So once we've measured these different concentrations, we're able to create profiles that are unique to cancer cases and controls and compare them between the two to see if they're different. And then using questionnaire data, I identified other factors that are linked to an alteration of these profiles. And so our approach was quite straightforward, actually. We used a prevalent case control design to select cancer cases and then age and sex matched controls on a 1-to-1 ratio from within the Atlantic Path cohort. In addition, we selected a number of participant information variables from the questionnaire, including family history of cancer, smoking frequency, physical activity, etc., because these variables were either associated with cancer risk or because we thought they might influence a methylation capacity. And from the toenail samples, we were able to use a novel analytical approach to extract the total metal concentration found in the toenails and the different species of arsenic that were present. The implications of this work are that the risk of chronic exposure to arsenic in drinking water may occur at lower levels than we previously thought. Like I said, arsenic in Atlantic Canada is higher than we'd like, but it's not the highest in the world. And so there should be potentially, with further research, a reevaluation of the World Health Organization and Health Canada recommended limits of arsenic. And in Nova Scotia and the other Atlantic provinces, there should be improved monitoring at a governmental level of private wells, because right as it stands now, private well monitoring and testing falls on the well owner, and their compliance is quite low. So people don't know if their arsenic levels are elevated. And finally, the big take home from this is that arsenic methylation is a better predictor of cancer than total arsenic. And the use of toenails really provides a promising option for an upstream biomonitoring project, similar to what Jacob has just described, where we could profile arsenic levels and then assess one's cancer risk with the generation of future studies. And that's all I have for today, and I'll accept any questions.
That was great. Thank you very much. So just as before, if anyone has got any questions, we've got a few minutes left. Please type them into the Q&A now. I have a question, I guess. So one of the interesting that there was no significant finding with total arsenic, and it seems to be the sort of speciation or methylation capacity that's important. How limited is this by the fact that I think you use prevalent cases as opposed to incidents? So these were cases where people had reported a diagnosis already at baseline when they were giving in their toenails as well.
That's a great question, and I appreciate that question. We'll hopefully be able to address this, or colleagues that come after me will hopefully be able to address this exact question as we use incident cases in future analysis. So this project will be expanded. But I would say that previous studies have also found that methylation capacity is a better predictor. Although previous studies in other areas have also been assessing much higher arsenic levels in total. So I think that it's a byproduct of having relatively lower arsenic levels compared to areas like Bangladesh and Taiwan. And also the fact that these are people who have previous exposure to, they have a previous cancer diagnosis. So hopefully that future studies can really clarify any association there.
But your take-home message is that you think that the 10 micrograms is probably potentially too high, or it needs reassessing.
Yeah, given more time, I would delve into that a little bit more. But basically what we think is that 10 micrograms is low, but it's potentially not low enough, and that just even chronic exposure at a very, very low level can reduce one's ability to methylate arsenic, and then the process becomes even worse. So you're exposed to even higher levels of the MMA species in your body.
Great, thanks. And so we have a question here. So were you able to link the toenail samples to specific regions of Nova Scotia? And did the toenail samples with lots of zinc arsenic come from heavy metal hotspot regions?
I think I'll need to get you to repeat the second part of that question, but with respect to linking to specific areas, we were not, although I did know what province the individuals were from. So I was actually sampling people from all four Atlantic provinces, and most of them were actually in New Brunswick, but we weren't able to tie them down geographically within provinces.
Okay, and so the second part was, did the toenail samples with lots of zinc and arsenic exposure come from heavy metal hotspots? So I think you sort of answered that.
It would be really interesting to do a geographic analysis to understand this association better, but previous work done with Atlantic Path has shown that the concentration of arsenic in water was highly correlated with the concentration observed in toenails. So I think the answer to your question is probably yes.
Yeah, no, I think that's right. And there has been some extensive work I've been involved in where we've been able to map the exposures, and there's regional variation, and it ties in very closely with the geology. Okay, another question. Is there any link between high fish consumption in the Atlantic provinces and the high arsenic levels?
This is a question I get quite frequently, actually. And the best answer that I can give you is that, of course, ingestion of foods with arsenic in them will contribute to your exposure. But the common thought right now in the literature is that the form of arsenic present in fish and other foods is organic and is not as bioreactive and bioavailable to the body. So your body is quite efficient at excreting the arsenic that you ingest in fish in particular.
Great. I think we have one more question. I think we've got a couple of minutes if we've got time. So what are some limitations with using toenails as a biomonitoring matrix? Limitations.
Well, I think that toenails are a better solution than urine, but one limitation that I encountered is that very few studies have used toenails at this time. And so it's difficult to compare your results to what other people have done, because what your body excretes and stores may be quite different when I'm talking about toenails versus urine. Another one is that we would really like to be capturing exposure over the life course. That would be the best case scenario. And while toenails improve upon urine, they're still only capturing about up to a year. Toenails are better than nails as they're not in contact with toxins in every day, but there are of course still potential for external contamination.
Now, I think then maybe the solution is repeat measurements, repeat sampling, and that would then be over the life course. That's fantastic. So we're just coming up against the hour, so I will call the this webinar to an end. Just like to thank the three speakers for providing a fantastic overview of really exciting master's and doctoral research, and some really good examples of using CanPath data to solve some pretty important questions and problems. Thank you all for the attendees as well. It was a great turnout. Look forward to seeing you all again virtually, at least, at our next webinar. So thank you very much for attending. Goodbye.